Thank you. So I treat skin cancers. Because of the virtue of skin cancers being mostly in a white population, I have no expertise in addressing racial disparities. I am part of the problem that we are faced with in terms of rising healthcare costs. I originally hail from India, and I was a direct witness to the complete, utter lack of resources when it comes to providing basic healthcare. My medical scientific interests led me here, and now I'm involved in testing and prescribing cutting-edge immunotherapies, which also happen to be super expensive. And because of those therapies, our patients are living longer, they are getting more treatments, but that's also contributing to rising costs in healthcare. And we are still not able to predict which are the patients who are most likely to benefit. So one of our talks today is about precision medicine, and so I look forward to hearing that talk about how can we identify patients more likely to respond in a better way. And second of our talks is about biosimilars, and I hope to be educated about what are biosimilars and how can they contribute to solving some of the problems that we have. So it's my great pleasure to introduce Dr. Hoifung Poon and Dr. Gary Lyman. So Binay, just tell me, do we have slides coming up one by one, or?
Yeah, so the panelists will be on the stage.
Okay.
And we might have to look at it.
Okay, perfect. So please have a seat. And so Dr. Poon is, I understand you have a connection with the University of Washington, and now you are at Microsoft, and you are focused on using artificial intelligence towards the pursuit of precision medicine. So it's my great pleasure to introduce you, and thank you for being here.
So I work for Microsoft. I own Microsoft stock, so I don't know what other, I don't have any other thing to disclose. So like I said, I mean, you guys are the expert in all this, so this should be fairly sort of common sense for this crowd. But for me, like the first time when I get exposed to this kind of data, it's kind of pretty shocking. So for example, the left, you can see a graph where here are some of the top drugs by growth cell. Under each drug, the blue person signifies the person who will respond to the drug, and the red person signifies the one who would not respond, right? So you can see red basically completely dominate the blue. So obviously the well-known problem in medicine, the trouble is just like how can we tell in advance who will be those who responds and not, right? So for example, in cancer, we used to know the originating organs. We can look under the microscope, look at the shapes and size and so forth, but there is a limitation in distinguish the cause for the cancer. And something like this kind of sensor technology really holds great promise because, for example, cancer is caused by mutation, and having the tumor genome is really like we are staring literally at the source code for the cancer, right? So we just need to figure out how to interpret them. So this is kind of like sort of another wow thing when I first encountered it. It's almost like a miracle, right? So on the left is this sort of like late-stage melanoma cancer patient, and the standard chemo doesn't work, but if we actually target it, if we apply a drug that specifically target this BRAF gene mutation that is in his cancer, the tumor almost miraculously melted away in less than 4 months, right? So that's really sort of the dream scenario we want to get at, but obviously we haven't quite solve the problem yet. Cancer is very robust. Eventually figure out a way to come back. So we are sort of like kind of like at the very exciting sort of crossroad, sort of like inflection point where we caught a glimpse of the exciting future. The question is how can we sort of move it forward much more efficiently. So as I sort of look at, as I mentioned earlier, the challenge is that we have all this big data. They potentially sort of hold the information, but to some extent, to find the right information there, it's kind of like looking for the needle in the haystack, right? So on one hand, for example, just interpreting those individual pieces of data, like figuring out what are those genes originally do, are those mutation functional, any sort of the approved or on trial treatment might be applicable, right? So that is kind of become sort of like what I would call the knowledge bottleneck. But increasingly also the emerging opportunity is that not just considering like single drug mutation and gene, but actually consider a combo to, for example, get over those relapse and so forth. And here the solution space actually, you immediately get a combinatory explosion, right? So this is what I would call sort of the reasoning bottleneck is how can you sort of combine all this knowledge and data and move things forward. And the hypothesis is that artificial intelligence like machine learning or NLP might hold some promise to help actually advance this course. And I will talk specifically sort of grounded in a key use case of molecular tumor board, right? So tumor board have been around for a long time, obviously. The new part is sort of this molecular bit is like, given all this sequence mutation, how can we make sense of them and how can we make use of them, right? So we have this is one in UCSF and sort of a bunch of people hunker down in the war room, staring at this 400, 500 mutation, and let's see what can we do collectively to figure out about it. The challenge is that this is not scalable, right? So in the U.S. alone, there were 1.7 million new cancer cases, and Dr. Shah mentioned like globally that is a far bigger number, and to some extent, it's kind of ironic because we now have this fantastic technology like sequencing that we can apply to every tumor, but then only a fraction of them actually get sort of like utilized fully by this kind of tumor board. Like for example, in Memorial Sloan, they pretty much sequence everybody, amount to tens of thousands of patients every year, but their board can only review a fraction of them, right? So the hope is that can we sort of democratizing this kind of tumor board so that everybody can actually get the best of the kind of treatment consideration, even if we have infinite number of you guys like experts and time, economically this just don't work, right? So we sort of really need a way to sort of help dramatically empower this board so that they can actually review cases much quickly. So one of the kind of the sort of the bottleneck, knowledge bottleneck is what I mentioned earlier, is like, what do we know about collectively about those gene mutation and potentially applicable relevant treatments, right? And so this is a sort of a curation team, knowledge curation team in Memorial Sloan. Their job is basically to look into those PubMed articles and try to sort of curate relevant knowledge. There is only a small problem here is that every minute, the PubMed is a huge body of paper, right, 28 million already, but also every minute I'm talking here, there are 2 new papers popping up. Every day there are thousands of them, right? So doing knowledge curation against this kind of fire hose of new knowledge is very challenging. And an expert curator in one of our collaborators, they basically can curate only a few papers a day, right? So the challenge, so the sort of aspiration is that can we help them dramatically sort of improve such a knowledge curation speed? And one of the sort of relevant potential technologies, what we call machine reading. So it's just very similar to machine learning, right? You sort of teach the computer to reach certain competency of reading so that they can, starting from text like some sentence or paragraph from PubMed paper, and then automatically curate the structured information there. So on the right, you can see this is a genetic network, right, regulatory network. Each node is a gene or protein complex, and then each edge is activation or inhibition and so forth. But in general, there are you can imagine any kind of structural knowledge that you want to curate, and specifically the things that are relevant to the tumor board is something like this, right? So in PubMed, you might see a particular cohort with a particular genetic mutations, and then later on, the paper described that they are treated by certain drug, and there is a certain outcome, and this kind of knowledge actually, from this kind of text, you might start to infer that if there is a EGFR mutation specific then gafetinib might be relevant. So it's this kind of knowledge you want to get out automatically. So that's kind of sort of inspire what we call Project Hanover, and the one big piece of the technology we're trying to develop is that how can we start from text, right, and apply machine reading to actually automatically curate a lot of those knowledge to put it in the knowledge base, and those knowledge is sort of like kind of inspired by the sort of the clinical decision making directly relevant for those. And I would talk a little bit about how do we sort of get by some of the machine learning challenge in this paradigm that we are pushing on called knowledge-rich machine learning. And also the beauty of this is that we don't have to really get to 100% accuracy at the get-go, right, for machine reading. And that's actually not quite there yet. But what we can potentially achieve is that sort of do 99% of the legwork so that we can curate a set of sort of like plausible kind of extraction candidate so that the curator can look at them and very quickly just verify whether each one is right or wrong. And also the beauty of that is that so initially, this tool could probably potentially already increase the curation efficiency, right? Because right now the baseline is just so slow. But also by using the tool in this kind of assisted curation paradigm, then they basically each decision they make basically can feed back to improving the machine reader continuously. So that's kind of like our sort of overall project direction. Now, why is this problem hard? Why is machine reading hard, right? So here is one of the challenge, right? So turns out that people are very creative in figuring out different ways to say the same thing, right? So here are just a few ways to say that this tumor suppressor can inhibit this apoptosis pathway, right? So you have, and if you just look at a small corpus of a few hundred abstracts, right, and you count it, how many different words can be used to describe this kind of negative regulation, there is obviously a very long tail. So it's very hard to sort of kind of like directly, imagining, writing rules or something to try to do this. Also, a flip side of this is that you have seen before, like different kind of expression might mean the same thing, right? A flip side of the things that is that the same mention, same expression might mean completely different things, right? So it's actually pretty amusing when I find out that there is a gene called PDF, right? So now imagining, for you guys, it's probably not hard looking at the context to figure out this is actually a gene, not a document format. But for machine, imagining that's a kind of daunting challenge for machine is to look at each PDF occurrences and trying to figure out, is this a gene, is this a document format? So those two are kind of fundamental challenge in NLP in how to interpret the text. So one of the sort of really powerful tool is machine learning, right? So it's very hard to sort of try to sit down and write rule to do things because you need probably hundreds of thousands of rules, and also each rule actually have lots of exceptions, right? So it's very hard to do that, but you can potentially sort of teach the machine to automatically write a program for you, right? So that's essentially what machine learning is about. It's like you start from some input, and then you try to come up with some program automatically to produce an output, right? So for example, if you care about automatic driving cars and then you want to show a lot of the cameras and laser pathfinder that is observable to human drivers, and then how do the human actually drive the car, right? So in machine reading, for example, your input will be the text, and then your output will be the structured kind of knowledge that you care about, right? So this is kind of like a standard paradigm. Like I mentioned earlier, is that you can just gather a lot of this kind of input-output example, right? Imagining writing a program for automatic driving car, that's very, very hard, right? But you can just have a human to drive many, many hours, right? And then have all those input and output, and that can sort of feed into this supervised learning paradigm, and then the computer just figure out the program by itself. So, and these days you have heard like deep learning is kind of like everywhere, you can't escape it, but it actually have a sort of a very humble origin, right? So it's really back in 1940s, people actually inspired by the actual neuron, and they come up with this kind of mathematical sort of formula where you imagining your input is a bunch of real number, a vector, right? So you represent your input like those cameras or those laser pathfinder, you represent them as a vector. And now you will basically do a weighted sum over the input, right? And then you do a threshold unit, right? So if it's above some number, you fire, right? So you will output a 1, otherwise you output 0. So that's sort of inspired by how the actual neuron work. And so sort of the early excitement is that we figure out how to train this kind of stuff if we have input-output examples, right? But turns out that this kind of representation is very, very limited. It cannot represent anything that are nonlinear, right? So even really simple things that it cannot represent. So this basically sort of the first deep learning wave, and that almost immediately died down. The second wave is actually in the 80s when people realized that, hey, if we actually add a hidden layer, right? So not just input and output, but actually add another layer in between, we can actually represent way more things. In fact, you can argue that we can represent almost everything, right? So the challenge is that how can we actually, again, figure out a way to automatically learn those weights, right? So that's specify the program, essentially. So the second way, basically, you figure out there is one algorithm called backproc. Some of you might have heard of it. It's actually a very, very simple algorithm, and it can actually learn it in principle. But if you actually start to add more and more hidden layers, then this algorithm start to break down, right? So that is kind of like the previous kind of end of the deep learning. And now that's sort of really the latest one is actually the sort of the modern reincarnation of neural network. And the secret sauce there is that we have a bunch of new algorithmic insight that allow us to actually train a sort of very, very deep network with no problems, right? So that's one of the reasons why it actually become practically successful now, including training things for driving cars, right? But also another big reason is simply because we have internet, and we can now create very big this kind of input-output data set, right? So for example, how do you recognize an image, right? How can you recognize cat and dogs and so forth, right? The secret sauce is that somebody using Mechanical Turk to create a data set with 15 million labeled images, right? So that is impossible before the internet age. And also, because of the Moore's law, so now we have GPUs and other really fast computer that can actually, munging those big data and churn out the program. So that is kind of really behind all this deep learning kind of excitement. And one of the reasons for this, why we want multiple layers, is because this is a very efficient and compact way to represent lots and lots of variations, right? So imagining all the kind of different kind of phases that in the world, and they actually, if you throw it into a deep neural network, it can actually learn this very interesting sort of layer representation, right? So you can start to see sort of like lower level capture the edge detectors, right? And then they will become to compose things like parts, like the ear, the eye, and so forth. And then the upper layers start to capture things like different kind of faces model, like male versus female, and so forth, right? So by having this kind of sort of deep architecture, you can capture lots of variation in very, very compact way. And so that's really the one of the big power for deep learning that is really relevant to the challenge we have seen earlier, right? So you have seen the image, but think about the natural language challenge is a similar thing, right? You have lots of variation that you have to sort of represent and model, and that's why deep learning is such a promising approach. But on the other hand, the bottleneck, it creates sort of a new bottleneck. And this is not unique to deep learning. This is universal for all supervised learning paradigm, right? And the challenge here is that in things like biomedicine, it's not very easy to create sort of the desirable input-output examples, right? So you cannot turk a CT scan very easily to ask average person to label whether this is cancer or not, and so forth, right? You cannot, recruit Tucker to label PubMed paper for you, right? So they can distinguish cat and dog, but probably not about genes and cancer drugs. So that's kind of like the fundamental machine learning challenge that we need to try to tackle. And this is sort of the new paradigm that my collaborator and I have been trying to pushing on, is what we call the knowledge-rich machine learning. And the key idea is actually very simple, is that if you think about biomedicine and any other high-value verticals, right? So on one hand, it requires deep expertise to actually annotate those data, label data, so that's sort of a bottleneck. But on the other hand, because of this such an important domain, there are already a lot of rich sort of resource already available. So all those kind of ontology about genes and disease and so forth, they are up for grab, right? And there are also a lot of sort of manually curated knowledge base, which are incomplete and quickly went out of date if you don't maintain it, but they actually provide potential sort of resource that can help us basically sort of to some extent hallucinate sort of noisy training examples. And then with the so you can think about it to some extent is that we're using those knowledge to go through those unlabeled texts, right? 28 million PubMed texts up for grab. You kind of automatically label tons of them, and then you can train whatever your favorite machine learning algorithm, including deep learning, right? So that's kind of like our game plan. So we have done a bunch of work in different areas. At the beginning, we focus a lot on fundamental biology, like gene regulation network, genotype, phenotype, and more recently, we've sort of focused more on this kind of actionable, precision cancer treatment for tumor board, and also some of the open science stuff. So let me give you a few sort of cartoonish example to see how this thing works. So for example, if you care about how genes regulate each other, right? So that's a very important kind of piece of knowledge. So a bunch of years ago, Nature Publishing Group kind of teamed up with NCI to say, hey, those editors, right, can you guys spare some time to basically read the papers and curate this kind of regulatory network? So they created this fantastic knowledge base called the NCI Pathway Knowledge Base, or PID, right? So each, you can think about it as a like table at some level, right? So each row basically said, talk about one regulatory knowledge, right? So for example, TP53 can negatively regulate BCL2. It's sort of like what the role is about, right? The challenge is just that this editor, they create a very high quality knowledge base, but they bail out after 2 years because it's too labor intensive, right? And then the knowledge base quickly go out of date because it's just too many papers come up. But then the idea is that can we actually leverage this as a free lunch to actually sort of like conjure up a training example, right? So for example, if I already know that there is this piece of knowledge between these two genes, right? Can we do something about it? So the idea is that if you look at the unlabeled text, tons of unlabeled text, chances are some of this knowledge originally come from some of those texts, right? We just don't know where, right? If we know where, then we already have the label, right? But the problem is that we don't. But then can we automatically nail down some of the candidates for those, right? So for example, if we saw that TP53 and BCL2 actually co-occur very close by, then we can start to hypothesize that maybe this sentence is talking about this particular relation, right? So then we can say, okay, in this case, I will basically treat it as a label example, right? So now with this paradigm, we can very quickly annotate lots of label examples, right? Obviously, this is not foolproof, but all you need to do in machine learning is that you don't have to have every example to be correct, but overall, it just need to be sort of sufficient statistical signal to do the right things. So this is kind of one example, and then back to our friend of PDF gene, right? So how can we apply sort of generic other type of knowledge to help resolve this mystery, right? So at the beginning, each of the PDF, right? It could be gene, it could be something else. So you sort of have a binary uncertainty, right, at the beginning, right? But one thing to realize is that those PDF actually appear very nearby. So this is actually, you can invoke a sort of a linguistic knowledge, is that sort of a linguistic phenomenon called co-reference, right? So those PDF in one paper maybe mean very differently from something in another paper, right? But the PDF that occur within a paragraph or within very close by in an article probably actually all mean the same thing, right? So they are all correct, just like Barack Obama and Mr. President and they are sort of co-reference, right? So now with that generic linguistic knowledge, we can already dramatically cut down the uncertainty because we now know all these 4 PDF probably are tied together, They are either all refer to gene or not, right? And then another piece of sort of linguistic knowledge you can start to invoke is that often the author will actually first spell out the long name, like peptide deformylase, right? And then followed by this sort of acronym, introducing the acronym at the beginning, right? So this is another sort of well-known linguistic sort of construct called apposition, right? So you saw a long name followed by a parenthesis, acronym, right? So then you can infer that most likely the PDF actually means peptide deformylase, right? So now if you just combine these two generic linguistic knowledge apply to all this kind of instance, you can start to infer that all these 5 references probably mean the same thing. And then the third thing is that the heuristic that you see that PDF is very ambiguous because it's very short and it has many potential meanings, right? But peptide deformylase is a long form and it doesn't match a lot of other things that at least I'm not aware of, right? So that means that peptide deformylase, when you go to the Hugo and you find that, oh, this is actually the name for gene, and then this is very likely to be actually meaning the gene. So now if you combine all this knowledge, then you can actually start from the beginning where you have lots of uncertainty about whether PDF is a gene, down to the level that you think all these are actually really gene reference and to this specific Hugo ID, right? So I'll skip this example, but basically that's kind of like, that gives you some intuition how we approach this, right? None of those rules and knowledge are foolproof, so we model them with probability, right? So you don't need to sort of like know much about this, but essentially the underlying idea is very simple. So think about each of this variable, Z, Z1, Z2, Z3, right? Think about each of them is basically a question mark like, is this PDF a gene or not, right? So each of those Z at the beginning is kind of like a latent variable, something that you don't originally know, right? But then what this kind of probabilistic logic allow you to do is to introducing those top things like the F and G. Those are kind of the knowledge, right? So that you can introduce, that can instantiate in this specific case. And then what all those factors do is that they can define a probabilistic distribution. So at the beginning, the Z1 can be 1 or 0, corresponding to PDF is a gene or not. And at the beginning, you basically have a uniform belief, right? You don't know whether it's a gene or not, right? But by imposing those knowledge, then that can quickly sort of concentrate the probability to the cases and to where you almost kind of having probabilistic label for those for those PDF cases. And now with those probabilistic label, you can then train all your favorite stuff, including deep learning, right? So that's kind of like how we do things. We also explore some other thing about, I will probably skip this in the interest of time. But the point here is that we are also trying to augment the deep learning machinery by incorporating more and more other free lunch, including like linguistic analysis, like syntactic parsing, right? So for example, a sentence, we have lots of research and technology in decomposing it into like a tree, like noun phrase, word phrase, and then noun phrase decomposed to article and the nouns and so forth, right? So if we incorporate all those structure, then we can potentially help the learning, right? So fundamentally, machine learning is no magic, right? So the more of the signal you can cram into it, then the better they can learn. If you don't have any signal to give it, it will not learn anything. So that's kind of like our technology side. And here is sort of like our roadmap. So our most sort of kind of semi-mature effort is in this precision cancer knowledge curation side. So like I said, this is sort of like a protocol that we're still developing where we can imagine this as sort of assisted curation tool, right? So you care about certain drugs, and then we basically summarize what are the genes that the machine reader think there might be some relevant facts, and then you can start to see actually drill down to individual pieces of the facts, right? Many of those facts might be wrong, many of those extractions might be wrong, but the key is that by having this kind of interface, when you highlight those entities and so forth, then the knowledge curator can very quickly to say, hey, this is actually a right extraction, right? This is what I like to put into my database, and this one is actually error, and it could be a gene error, and so forth. And so at the get-go, hopefully we can already help them curate much faster, but also throughout the process, they can gradually improve. And then another sort of, so these are sort of, here the text is like the literature, the papers, right? But then there are also lots of text, like, depending on how you count, like 70–80% of the information are in the electronic and in the EMR, right? So one of the bottleneck in actually using EMR information is that those texts, those contain rich information, but they are really locked into the text, and the machine cannot very easily process them, right? So the question is that can we actually, using the similar machine reading technology, to unlock those, put them into sort of structured patient information, right? And also, if you look at things like clinical trial, all 20,000 trial, you can download them. And if you look at there is lots of text here as well in the, for example, eligibility criteria, right? So that specify who should be enrolled and not, right? So if you sort of combine the two, and one observation is that a lot of trials actually fail because we simply don't find enough of the patient, right? So right now, a lot of this is done by word of mouth, right? So if you encounter the right physician with the right, knowledge about a trial, then you're lucky. Otherwise, so for the patient side point of view, this could be actually a life and death challenge as well, right? So one of the sort of the aspiration down the road is that can we apply the similar machine reading technology to understand the EMR sufficiently in the text, understand the eligibility criteria sufficient so that we can dramatically facilitate matching, right? But also, ultimately, I think the most exciting prospect is actually how to address what Dr. Sharp pointed out, is that why we are not using EMR in a much better way, right? So if you look at all the cancer patients, right, and then count how many of them actually get enrolled in the trial, so the number might differ, but some of the number, but the consensus is in single-digit percentage, right? So vast majority of the cancer patients are treated outside of the RCTs, and in a lot of those cases, the standard of care doesn't work. And then the oncology, in each time, basically, like you guys probably invoke sort of your prior knowledge to try to apply certain sort of drug that may not be, approved for this indication yet, right? So the off-label drug use case. But if you think about a lot of them, they are actually a huge treasure trove, like sort of like all this N of one trial, right? So that's kind of like 97% versus 3%, right? So lots of statistical power there. Obviously, you need to figure out the confounders and all those things, but still, the question is that how can we sort of unlock those information, and so we are starting doing some baby step kind of towards this aspect. Is like, can we sort of understand the EMR well enough to extract some of this kind of off-label drug use so that can contribute to this goal, and eventually sort of treating the whole EMR as a big virtual trial. So you can derive lots of benefit from it, right? We can cut down the drug development cost potentially, and so forth. So that's kind of like the overall sort of project handover, kind of like a few sort of key use case in mind. I want to acknowledge my collaborator, without whom obviously I cannot do any of this. Thank you. Okay, sure.
Thank you. Thank you, Dr. Poon. So our next speaker is Dr. Gary Lyman. He is a member at the Fred Hutchinson Cancer Research Center. He's actually the co-director of the Hutchinson Institute of Cancer Outcomes Research. He's a medical oncologist who is focused on breast cancer patients, and also a professor at University of Washington. He's going to be talking to us about biosimilars in oncology. Thank you.
Well, good afternoon, and thank you, Dr. Shaw, for the invitation and for all of you sticking with us through the afternoon. It's a pleasure to be here. I was asked to talk about biosimilars in oncology, And I'm going to make the presumption, I do apologize to those of you familiar with this topic or have even spoken on the topic, that many of you are not familiar with it. So I'm going to lay the basics, take you through it, and why we're dealing and excited about this issue at the present time. To weave it into the rest of this meeting, I will start off by saying the fundamental driver for biosimilar discussions, yes, there's competition, yes, there's marketing issues, but it's cost. It's the rising, and I would say unsustainable rise in healthcare costs that we'll hear a great deal. We've already heard quite a bit about, we'll hear more about through this conference today and tomorrow. Biosimilars are not the end, but a means to an end. That end is reducing cost, improving access to care, improving the quality of care, and improving patient outcomes. So keep that in mind as we go through this. There's a lot of regulatory issues here. There's a lot of new things coming, but it's important to keep that in context. So starting off with your question, what are biosimilars? The FDA actually, well, if I can go back here, the FDA actually formally defines a biosimilar as a biologic product that is highly similar to a U.S. licensed reference biologic product. Of course, this is a U.S.-centric definition, for which there is no clinically meaningful differences in safety, purity, or potency of the product. So keep that definition in mind as we go through. But why are we talking about biosimilars today? As I said, at the bottom is cost. So we know quite a bit about healthcare expenditures. Again, you've heard already a fair amount about it. Over 1/3 of the spending in the US is concentrated in 5 therapeutic areas, and right at the top is oncology. We also know that cancer drug prices and the cost of cancer drugs have risen more rapidly than the overall care of cancer patients. And together, they've risen far more rapidly than overall healthcare expenditures in this country, and all that, of course, more rapidly than the gross domestic product. So it is a rapid, escalating problem, the cost of cancer care. You can see a big part of the problem illustrated by the top 10 Medicare drugs as of a couple of years ago. I don't think it's changed too much. And if you go down these, you'll see many of these are oncology drugs, and many of them are the new wave of exciting biologic therapies, and that's where our focus will be. And just you see the cost in terms of millions or in some cases billions of dollars. So over the last, the period between 2010 and 2015, about a 5-6 year period, we see that the total cost for cancer medicine in the US has risen, is essentially doubled, almost doubled, increased by some $16 billion. And we can look at that a big part of this cost is the oncology product itself. A part shown at the top here is the supportive care, things like hematopoietic growth factors and antiemetics and other supportive care agents. But the biggest part of the growth are on the cancer treatments themselves, and we'll come back to that issue. The group at Memorial have highlighted in a previous publication what has happened to the monthly and median costs of cancer drugs at the time of FDA approval. And of course, you can look along for several decades, and then all of a sudden, in the late 90s and early 2000s, a rapid and steep rise, where is at the present time, in recent years, the average cost of biologic therapies at the time of approval has been averaging around $10,000 a month, and that was mentioned earlier as well. This just illustrates that a lot of that growth in biologic therapy or in cancer therapeutics is the targeted therapies. So that light blue area represents the growth in late-phase oncology products. So these are products in late development or at the time of approval. And of course, there are other things. There's traditional cytotoxics. They haven't changed very much. And radiotherapeutics and so forth. But the vast bulk of this are the targeted therapeutics, the monoclonal antibodies or small molecules. Globally, and we are a global conference, this is even more concerning to see the rate at which the growth of global biologic sales has increased over this time between early in 2000s through the past year. So the cost of the sale of biologic therapies has increased tremendously over less than 2 decades, about a decade and a half. Dr. Prasad, I think, is in the audience. He has broken out in this piece in Nature Reviews a comparison with one of our Scandinavian developed countries. We heard about Scandinavian countries this morning. This one is Norway, but although the average salary is adjusted, of course, for different currencies, is essentially the same between the US and Norway. Look at the difference in the cost of 1 month of treatment with the top 10 best-selling anti-cancer drugs, the US versus Norway. So it's quite startling, of course. So why does this matter to us? I think it's obvious. We obviously have patient advocates, providers in the room, and just people, most of whom, including myself, have had family affected by cancer. Why this focus on cost? Well, first of all, there's a huge variation. If you look at geographic, if you look across ethnic, racial, financial variation in the population, you see a huge variation in the cost of care, and I would dare say in the quality of cancer care. This will be highlighted this next week, and you'll hear more about this tomorrow as we issue the first community cancer report looking at quality and cost throughout the Western Washington state for the first time next week. Importantly, patients bear an ever-increasing part of that cost, what we are calling the financial burden, the financial toxicity, whether it's copays or out-of-pocket expenses. Patients are being left with more and more, and assuming they have some insurance to begin with, and many, of course, are underinsured. But even if they have insurance, increasingly, a larger and larger fraction of that cost is being passed on to the patient and, of course, to the family. Again, the importance is cost affects access to care. It impacts on treatment decisions, whether to start treatment, continue treatment, stop treatment early, and after treatment, it can have an enormous impact on the patient and the family. We've shown increased rates of bankruptcy. We've shown patients with cancer who go into bankruptcy, which is at least twice as high as non-cancer matched patients in the Northwest, have a higher mortality rate. So it really has downstream effects that we all need to be aware of. Not to give Dr. Prasad too big a head, here's another reference which I think really greatly simplified some other work I saw on this topic, but this really hits it home. Looking in this case at the median monthly household income, this red line that has been essentially flat for several decades, and the blue line, which again, late 90s, early 2000, representing the median monthly cost of new anti-cancer drugs in the US. And it's continued up. And of course, there are projections that would say this is approaching exceeding the median income of families, and could well exceed that in the coming years. So the cost of care, health care in general, and cancer care in particular, is rapidly outstripping our ability to pay for it as individuals. So what are the potential benefits from biosimilars in such a system? Well, as I mentioned, these are hypothetical, and you'll have to decide at the end whether, in fact, it's a realistic hope that the biosimilars will have these effects. But one is, of course, greater access, improved affordability, greater proportion of eligible patients being able to get these exciting new therapies, reducing economic disparities. We're all excited about these drugs, but if they can't be afforded and they can't be taken, they're not going to have the intended impact. Greater competition, of course, that's part of the equation. Increasing competition driving down biologic costs. And in a market-driven healthcare system, this may work, yet to be shown. Biosimilar manufacturers also can take advantage of the latest technologies. So as these biosimilars come on, there are new technologies that come with them. And then, of course, we would like to think at the same time, we're not going to discourage, but actually foster innovation and new therapeutic approaches. Again, yet to be shown. So just a brief discussion of the science of biosimilars, so we're kind of all familiar with these. These are quite different. The biologics are quite different than the simple chemical drugs. I illustrated here with aspirin. A small molecule, low molecular weight, well characterized, produced by chemical reactions in the laboratory. You can make identical copies of these agents. They're completely characterized, they're relatively stable, and for a large part, they're non-immunogenic. They don't produce antibodies. On the other hand, you take an example of a biosimilar, a monoclonal antibody. You see it's a much larger molecule, a much larger, higher molecular weight. It's complex, it's heterogeneous, it's produced in living cells or living systems. It's impossible to ensure identical copies of such complex molecules. It's impossible to fully characterize their composition. They may be unstable and very sensitive to variation in external conditions. And they may be, the potential is there for immunogenicity or antibody formation. So how are we dealing with these new biosimilars that have just in the last couple of years entered the marketplace in the US? They've been in the market in Europe for some 10 years, and I'll come back to that in a minute. But, so on the left, you see what traditional biologic therapy approval process is. You start at the bottom with molecular characterization. You do preclinical studies in the laboratory or in animal models, and then through a series of phase clinical trials, phase 1, 2, and 3 that you're probably all familiar with. After approval, there may be or may be not a recommendation for post-marketing surveillance for that rare, delayed, or unexpected toxicity. On the right-hand side are the biosimilars. Now, if we required the same amount of work for the biosimilar as the original biologic, of course, we're not going to save any money. It's going to cost just as much to develop, and because costs are going up, it may even cost more to develop the biosimilar than the original. So the FDA, in their wisdom, and this is with a lot of public input and scientific input, have said, let's put the focus on molecular characterization to make sure the amino acid sequence is the same, the structure is essentially the same. Preclinical work, the PK, PD, pharmacokinetics, pharmacodynamics, it behaves the same. We look for immunogenicity and make sure it's not going to be a major problem. And then provide some clinical data, but not these multiple large randomized trials. And then we'll put another focus after approval, once we're sure, as we can be, that it's safe and efficacious, on this post-marketing surveillance. So it's a flip in a sense, and it may be a little simpler to look at here, a forthcoming paper we have in the New England Journal. You start at the top with analytic study, looking at structure and function. You look at animal studies, PK, PD, immunogenicity, toxicities, and then some clinical data, again, looking at those elements. And then if there's still some uncertainty, about the safety and efficacy, you can then require comparative clinical studies to determine equivalence to the original biologic. So it's kind of a reverse pyramid or flip pyramid. So what about integration into practice? As I said, we already have some of these in place. A few weeks ago, we published a statement from the American Society of Clinical Oncology, primarily in an effort to educate oncologists as well as other providers of cancer care throughout the country and throughout the world, and also educate our patients to the extent they want to go through a document of this sort. So this is, was recently published in the journal Clinical Oncology. As I mentioned, Europe has been doing this for the past decade. Their first biosimilar was introduced in 2006. There are now more than 25 biosimilars approved in the European Union. None has been withdrawn or suspended because of serious side effects or a lack of efficacy. And therefore, over the past 10 years, the EU monitoring system for safety concerns has not identified any relevant differences, as it says here, in the nature, severity, or frequency of adverse effects between biosimilar medicines and their original reference biologic products. So in the US, the top here shows some of the non-oncology biosimilars that have been approved. You may recognize some of these. You may not. You might recognize the reference product on the far right-hand side. And the 3 oncology products that have been approved, only one of which is available at the moment. And we won't go into the reasons for that. 2 have been very recently, and there are multiple others in the pipeline at the FDA requesting approval. The one that is approved is a supportive care product, filgrastim Sandoz, or SNDZ. And there are now biosimilar approvals from the FDA for bevacizumab and trastuzumab and HER2-positive breast cancer. But again, they're not yet hit the marketplace in the US, but they will. So what about integrating into practice? We do a lot of clinical practice guidelines. When the biosimilar myeloid growth factor, GCSF, that filgrastim SNDZ was approved, there had been a previous agent that's a biosimilar in Europe, but it went through a regular approval process in the US, Tibo filgrastim. The ASCO guidelines and the NCCN guidelines rapidly looked at the data and said, these are all options to be used in patients with cancer. They seem to be similarly efficacious, safe, and can be used to prevent treatment-related febrile neutropenia. And the choice depends on convenience, cost, and other clinical conditions. However, there remain concerns. I talk to my colleagues all the time. I present at the annual meetings for ASH, for ASCO, other organizations, and I hear the concerns that clinicians have, in many cases trying to represent their patients. Physicians are skeptical of the limited clinical data behind these providing sufficient evidence of efficacy, safety, whether there'll be an impact on reimbursement. Of course, that's a practice concern that exists. And of course, clinicians in the US particularly are concerned that they're being forced to use something that they were happy with the old product as opposed to taking on something new. Physician perception is that in some cases that cost is the main issue here, and it is. The strong clinical data will be important for acceptance, and I think that is true. So in some cases, going light on the clinical data may not serve the drug or the company well that wants to produce these agents. They may want to put forward a fairly robust clinical data to convince their clinical colleagues that it is, again, efficacious and safe. And education, I think, is at the hallmark. So we all understand what these products are, what they aren't, what they can do, what they are yet to be shown to do, and this will be taking place both online and at national meetings. So finally, what are these challenges that we have to face? And we really just touched the tip of the iceberg. there is this need for post-approval pharmacovigilance. Again, limited clinical data at the time of approval. Approved agents, the FDA has the authority to grant extrapolation to indications for which the reference product was approved, but no clinical data was provided for the biosimilar. So if the biosimilar is approved for one indication, the FDA may grant or extrapolate to all the indications that the original biologic was approved for with data, even though the biosimilar has not provided similar data. Product variability and drift. This is something we haven't thought too much about. It's very true of biosimilars, but it's also true of the original biologics. As minor changes in manufacturing take place over time, you put in a new chemical, you open a new manufacturing plant. There may be changes in these molecules that need to be examined. And the FDA is very diligent about every time there's a change in composition or in manufacturing to go examine the product that's coming out and make sure that it still seems to fulfill the definition of highly similar to the original biologic. Also making sure there's no immunogenicity and so forth. There's a new concept, I won't go into details, called interchangeability or automatic substitution. This is a new level, a higher level of approval, so a biosimilar can get approved as a biosimilar and go on the market. But if they want to get a label as interchangeable biosimilar, they have to provide even more data showing that switching back and forth between the original biologic and the biosimilar doesn't make any difference, has no adverse effects on efficacy and safety. To date, there's no such biosimilar in the US that's had that designation. So again, we have this reverse pyramid where it starts with characterization of the structure and function of the molecule, animal studies, some early human studies, usually in healthy humans, checking for immunogenicity, and then some clinical data on efficacy and safety, and then finally post-marketing surveillance, which is extremely important. Extrapolation is based on what is thought to be a reasonable requirement for development and approval of biosimilars based on this structure. Time will tell whether clinicians and patients will be fully acknowledging of this. That extrapolation assumes the same mechanism of action, the same pharmacology, PK, biodistribution, the same expected toxicities, and other factors that might include the impact of comorbidities or concomitant medications on the drug. So based on this rationale, extrapolation is here. Again, variation and drift is something that is inherent to the biologics, including the biosimilars, but they're both subject to this, and this is being actively monitored, but it's important that we all be aware of it. This just lists some of the sources of variability so you understand. These aren't simple chemicals. They're very complex biologic products coming out of living systems, many places for variation. The immunogenicity issue, which may lead to loss of efficacy by binding to the biologic or the biosimilar, neutralization of endogenous proteins, or a generalized immune response, an allergic response, anaphylaxis in its extreme form. So again, that whole issue is, to date, we do not have a biosimilar that's approved as interchangeable. The definition of interchangeability requires more data. It's a higher level of standard, but it's now on the books. There is a process, there is a policy at the FDA for granting that. And to preempt this, we now know some 35 states throughout the country have issued have promulgated laws that would say you can't do that without notifying the clinician, the prescriber. Under the federal law, a interchangeable biosimilar could be switched by a pharmacist without any notification of the patient or the provider, the prescriber. So these states have taken proactive action in advance of a drug getting that designation of interchangeability and said, we're not going to allow for that without some notification of the provider, and so that the patient and provider can be aware. There are specific switching studies that are required by the FDA to get this designation, so it's yet to be seen whether the biosimilar companies will strive for that, but it could be a marketing advantage to them. So in the US, this is just the timeline to biologic products, patents that are expiring right up through the current year. So these are targets for biosimilar production. I've mentioned the bevacizumab, the trastuzumab, but many of these are already being targeted by companies to develop biosimilars, rituximab, a whole host of cancer therapeutics. And this is where the issue becomes even more complicated. When you're talking about supportive care, it's one thing. When you're talking about changing the patient's cancer drug, their actual cancer treatment, to one of these, the biosimilars, I hear concerns expressed. They don't want to be the first one on their block to jump and use these. So the real goal, of course, is the cost, again, the market for biologics or biosimilars in the US is anticipated to grow tremendously over the rest of this decade. So it's expected to grow well in excess of original expectations, but we shall see. The real question in my mind is whether these will gain traction in the US and over what period of time. In Europe, they have taken hold. They've become the dominant form of biologic therapy in many European countries. And again, no major issues have come out of that. US is a very different environment. And I think despite education efforts, it's yet to be seen whether these will rapidly take hold and replace reference biologics in the US. If they do, again, here are projections that there could be a cumulative savings of anywhere from 50 to over $100 billion through the rest of the decade. But, and this is for both the 5 major countries in Europe, the economic powers, and the US for 8 key products. So will biosimilars be accepted and utilized in the US? We don't know. I think they're here to stay. I think it behooves all of us to be very educated, understand, and inform our patients what they can do and what the remaining questions are. Will biosimilars help reduce unsustainable increase in healthcare costs? Again, yet to be seen in the US setting. And will biosimilars improve patient access and reduce disparities? To me, these are the big questions. We need to find that balance between regulation and authorization or clinical use, where we require enough to demonstrate efficacy and safety for patients, but not so much that we haven't begun to curtail the rise in healthcare expenditures. Thank you so much for your attention.
Questions for our speakers? Either the microphone is too short or I'm just too tall.
Yeah, we should switch microphones.
Go ahead.
I wanted to ask Dr. Lyman, Gwen Nichols from Leukemia Lymphoma Society. I think my concern about the biosimilars is not so much about efficacy, because I think the testing is rigorous enough. My concern is what we've started to see when new targeted agents in the small molecule space become generics. The price in the US is not dramatically lower than the price of the original drug. And given that the biosimilars are even more complex to make, and so the companies that are making them are often the same companies that are making primary initial drugs. Do you have a sense that there'll be any way for us to help the biosimilar cost be more reasonable so that we actually realize the savings that you're talking about?
Yeah, well, clearly, you probably can tell from my talk, I share some of those concerns in the U.S. marketplace. The encouraging news, again, to whatever extent you can draw parallels in Europe, there has been, at least in some countries, about a 25% lowering due to competition in the cost of some of these drugs. But those are very different health systems, very different forces or drivers involved. And so in the U.S., I think it's yet to be seen. So I agree. I'm not too concerned about efficacy and safety, but the U.S. is just a very difficult market. And as long as we are a market-driven healthcare system, and we can talk for days about that, yeah, I think it's going to be a challenge. My guess, if I have to put one forward, is at most it's going to bend the cost curve maybe a little down. It may continue to rise, but not as rapidly. We shall see. And the other thing is, the other encouraging thing is for some of the big spenders here, largely monoclonal antibodies in cancer therapy, there are multiple biosimilars seeking approval.
Yeah.
And if we have 4 or 5 drugs there, it may be enough of a force to bring prices down. It's not going to bring it down like generics do. That's just not in the cards. But if we can, if that will bring it down 20, 30%, we're talking about real money here that will help.
Thank you.
This also to Dr. Lyman. So we've done a lot of market research, primary market research, where from an oncologist perspective, there is more acceptability to biosimilars. They've overcome the fear over the regulatory process. I think they're still concerned about extrapolation. We've surveyed over 500 patients. But I think the most important piece is the relationship between payers and manufacturers, right? I mean, at the end of the day, when everything is equal, prescribers are not going to recommend therapy unless reimbursement is assured. And payers will make decisions based on their formulary, whether it's the biologic or the biosimilar. And some of this is dependent on the rebates between manufacturers and payers, which completely the prescribers are out of that. So I think sometimes we put a lot of pressure on prescribers where they really, I mean, the decision is probably not theirs, because if a payer tells you you are going to prescribe the biologic Avastin, you are unlikely to do the biosimilar.
Well, I mean, you get at the heart of where I think the problem is. I don't think it's the biosimilars the problem. I think it's our system of negotiated pricing, of dealing with insurance companies and struggling to make sense of access to a whole range of therapeutics in the cancer setting. The biosimilars, again, is one effort to maybe curtail costs somewhat, but we probably got bigger issues to face if we're really going to enhance patient access to the latest exciting cancer therapeutics.
I actually have a question for the other speaker, but
Good.
Just in follow-up to the current comments, I wonder actually if biologics, if that would not be the first place where we need to ask for cost containment or a political solution, as the drug development is so much cheaper, and yet they have about 85% of the drug price here in the US. That might not be a setting where we could say Congress mandate, any biologic, any biosimilar shouldn't cost more than 70% of the original price as the development was lower. But yeah, those are political forces that it's not worth discussing.
I'll tell you.
Yes.
I understand where you're coming from, and I think it's absolutely true.
But Hoifung, really remarkable what you're doing. The problem is in the oncology, if I may challenge you, community, we have seen, or I've seen 2 disappointments so far. There's Watson, and we actually get to see some of the consults where Watson just finds the clinical trials for the patients, and it's awful. It's essentially useless right now from what we're seeing from what I've seen a few cases now. How, do you want to say how you maybe are different from Watson? Because Watson is, in a non-knowledgeable person's view, seems to be the holy grail right now in AI. And secondly, so we have had this large clinical genomic cohort study at Duke where we were going to get the clinical data also through IT, electronic medical records and natural language processing. We had a phenomenal team. And it turns out all the money went mainly into IT, them trying to figure this out, and it's not possible. It wasn't possible. And talked to the kind of latest people in natural language and currently, and it's still kind of far away to do that. Again, do you want to comment on those two big issues? And I know actually the person who led the IT part at Duke, she's now running Flatiron, and they're doing hand extraction.
Well, excellent question, and I expect this will pop up absolutely. So thanks a lot for asking that. So I would say, first of all, I would say Watson, their initial success in the Jeopardy, right? It's a fantastic, remarkable engineering effort. And however, if you look at the Jeopardy solution, it is no different than the standard supervised learning paradigm. I'm actually very familiar with, we are in the same DARPA machine reading grant. So their slogan is like, let 10,000 flower blossom. So basically you throw all different kind of features inside, but it's fundamentally driven by all the past Jeopardy game as label data. So that's kind of like, that's kind of part of the difficulty when you like leaving the Jeopardy game and start to think about how can I take this kind of supervised learning paradigm to apply to some other domain when you don't have the label data, right? So go to HR blog or go to fashion design or go to biomedicine, and that's kind of like the major bottleneck. So to some extent, so that's sort of like where our research have been for many years, sort of like focusing on how can, like assuming that we are not going to have a lot of those label data, how can we figure out other way to compensate for the lack of it? So I would say that's kind of like one of the sort of the different emphasis. Now another thing that we are kind of trying to start sort of like in the sort of the small kind of like concrete step where we identify concrete sort of like pain point, like for example in those knowledge curation, right? So they're part of the current workflow to help the tumor board, but there is an obvious sort of value proposition there, which is that your baseline today is like curating 4 papers a day, versus if you can just simply double that or something, that will be sort of potential help there. So I think I don't have a sort of a magic answer in total, right? But I would say those two are the aspects that we are kind of focusing on. Now, Flatiron is actually a very, very interesting case, right? They are started by ex-Googlers, and we've imagining initially taking a technology and then it will be a piece of cake, right? But obviously that's not the case, right? So, and one thing it's kind of interesting is that they basically have sort of thousands of this kind of, sort of at least sort of like at the nurse kind of knowledge level to actually curating information from EMR. So for us, I think it's actually a very good sort of like validation in the sense that there is obvious value in those structural information, right? That's why Roche paid $2 billion for that, right? So the question is that can we sort of like make those curators much more efficient, right? So if we can, so sort of similar to our sort of like kind of precision cancer knowledge curation scenario is like, I mentioned sort of our Ceiling project in the EMR space. It's sort of like have many inspiration, but Flatiron to us is actually a great validation. Like there is obvious value there. The question is that can we just imagine just like double their curation productivity and or triple them, right? So.
My sincere apologies. We have run out of time, and I'll just encourage people who have questions to just ask the speakers individually. We need some time to prepare for the debate, and I appreciate your attention during this fascinating session. Thank you.